This is the day the Lord has made. We will rejoice and be glad in it. Woohoo! We get to start on chapter 25, the urinary system. I have this chapter broken up into 5 lectures. I'm learning how to do this better. And you have your schedule that I sent out to everybody. So hopefully that will help you stay caught up. As you listen to this lecture, as you read through your textbook, make sure that you have your learning objectives for sections 1, 2, and 3 written out, and maybe you can answer them as we go through the chapter and what's taking place. It might help you stay engaged instead of getting bored. So let's go ahead and start with the anatomy of the kidneys. Normally, I would not spend much time lecturing over the anatomy of the kidneys because we would have lab, and in the lab is where we focused on anatomy. But since times have changed, then I'm going to spend time on the anatomy. You should know the functions of the kidneys. So it's going to regulate water volume and solute concentration. It regulates ion concentration for the extracellular fluid. It ensures long-term acid-base balance. Remember, respiratory is short-term. Kidneys are long-term acid-base balance. Of course, this is the way we get rid of toxins, drugs, and waste. The kidneys produce erythropoietin, which regulates red blood cell production, and they make renin. Some people pronounce this renin. Anyway, this is part of regulating blood pressure. It activates vitamin D, and when necessary, it will make glucose. This is called gluconeogenesis. Gluco is for glucose, neo is for nude, and genesis is the formation of. So this is the formation of new glucose. Now, aside from the kidneys, the rest of the urinary system is really transport and storage. The ureters are going to move urine from the kidneys to the bladder. The urinary bladder is going to store it, and then when your bladder gets full and sends a signal to the brain, it tells the sphincters in the bladder to open up and dump everything into the urethra, and the urethra transports the urine out of the body to the external environment. So when we look at this picture of the urinary system, you can go back and look at this and see, hey, what are the things I'm supposed to know that's on my lab list that's in this picture? So you can see everything in front of the kidneys has been removed. The gland that sits on top of the kidneys is the adrenal gland. We talked about that in the endocrine system. All right. You can see where the renal arteries branch off the abdominal aorta and where the renal veins attach to the inferior vena cava. And you see how the ureters are long and go all the way to the bladder. And the bladder, let's call it the urinary bladder because there's another bladder that we have called the gallbladder, so you have to be specific. You can see where the urinary bladder sits in the pelvic portion of the abdominal cavity. And since this is the female system, you can see where the uterus sits on top of the bladder. So as a baby grows and the uterus gets bigger and bigger, the urinary bladder gets smushed. That is why pregnant women always have to go to the bathroom, is because the baby is smushing the bladder. And then from the bladder to the external environment is the urethra. I remember that the ureters go from the kidney to the bladder because alphabetically they come first. Ureter comes before urethra. And urethra is from the bladder to the external environment. Here's a cadaver showing the kidneys, the veins, the arteries. The ureter is the thin tube, the urinary bladder, and then of course in this you can't see the urethra. So let's look at the gross anatomy of the kidneys. Okay, these are going to be in the back behind all the intestines. And we're going to look at a picture in a minute that will show that they cover a space between the thoracic vertebra 12 and lumbar vertebra 5. The right kidney is a little lower than the left. As we said, the adrenal gland sits on top. There's a convex surface. It looks like a bean. So the outside curve is convex, the inside curve is concave, and it's there in the center of this concave place where we have what's called the renal hilum, and this leads to the renal sinus. The ureters, the renal blood cells, lymphatics, and the nerves are going to be located at the hilum. So there are 3 layers of supportive tissue. We have fascia, which anchors the kidneys. We have the fat capsule, which protects and cushions the kidney, and the fibrous capsule. And this is going to be on the surface of the kidney, and it helps prevent any infections from entering the renal tissue. The upper part of the kidneys are going to be protected by the thoracic cage and fat, but the lower part are exposed, and so anytime you get hit in the lower back, you can do kidney damage. A big indicator of kidney damage is going to be hematuria. Hematuria. Let me try again. Hematuria, which is blood in the urine. Anytime you see URIA, that's urine. Of course, hemat or hemato means blood. So let's look here. Inside the kidney, there are 3 regions: the cortex, the medulla, and the cortex is on the outside. The medulla is the middle, okay, and it's composed of what we call renal pyramids. The tip of the pyramid is called a papilla, and the renal pyramids are going to be separated by connective tissue called renal columns, and then the medullary pyramid and its connective tissue is called a lobe, and each kidney has about 8 lobes. The third part is called the renal pelvis, and your book says it's a funnel-shaped tube. That's about the best description you can think of. It's composed of minor calyces, which are closest to the papilla of the renal pyramids, and then the major calyces that collect urine from the minor calyces. The renal pelvis is where the urine will empty before it goes into the ureter. So urine is collected in the collecting ducts of the renal pyramid. It flows to the minor calyx, then to the major calyx, to the renal pelvis, and into the ureter. We have a picture here, and you should be able to identify these in lab as well as lecture. So you have the cortex, The cortex is where most of the nephrons are going to be. You have the renal pyramid. You can see it has a general pyramid shape. The tip of it is called the papilla. The papilla. Then you have the minor calyces here, here, and here. The major calyces. And then the renal pelvis. So when we look at this, the renal pelvis is going to be closest to the blood vessels, the area closer to the blood vessels. What are some of the diseases that we see with the kidney in general? One is called pielitis, itis infection, pielo, meaning the kidney. So this is an infection that has reached the renal pelvis and calyces. Oftentimes this is the result of a urinary tract infection. And then pyelonephritis, inflammation of the nephrons and the renal pelvis and calyces. A lot of times we just say it's an infection or inflammation of the entire kidney. Once again, the original cause is usually a urinary tract infection. This can damage the kidney. The kidney cannot regenerate itself. It has to repair. That means scar tissue, and scar tissue means loss of function. The renal arteries are going to come off of the abdominal aorta. They will separate into the segmental arteries, the interlobar, the arcuate arteries, and then the cortical radiate or interlobular. Okay, so there's interlobar and interlobular. Interlobular means within the lobule. Interlobar means between the lobes. I prefer cortical because it tells me that it's going to the cortex and to the nephrons in the cortex. Then draining from the nephrons, we have the cortical radiate veins, the arcuate veins, the interlobar veins, and then into the renal veins. Notice that there's no segmental vein. Once gets into the renal vein, it's going to flow into the inferior vena cava, go up the inferior vena cava and into the right atria. The nerve supply comes from the renal plexus, and you can learn more about that in graduate school. So let's look at the blood here. We're going to start with the renal artery. It's going to branch into the segmental arteries. The segmental arteries will branch into the interlobar arteries. The interlobar arteries will split into the, I lost my place. Interlobar will split into the arcuate and then into the radiate, the cortical radiate arteries. Okay. Ooh. And then you just play it backwards, except there are no, pay attention, there are no segmental veins. There's the arcuate artery. There it is. Okay. So you should be able to trace the flow of blood through the kidney. And this is a very, very nice little flow chart for you. Now's a good time to pause and to stretch. Draw a line in your notes because we're getting ready to go microscopic. Welcome back. Let's talk about the nephrons. The nephrons are the structural and functional units of the kidney. So if I ask you to define nephron, you will say nephrons are the structural and functional units that form urine. Each kidney has at least 1 million nephrons per kidney. Some have a few more, and yeah, they're amazing. Be thinking about, as you look at this, how God put all this together to clean and purify our blood of toxins and wastes and drugs. This is phenomenal. This is absolutely phenomenal how God put together this little tiny unit that keeps us up and running. So there are two main parts, the renal corpuscle and the renal tubule. Renal corpuscle and renal tubule. The corpuscle is composed of the glomerulus, which is a group of capillaries. Your book calls it a tuft. To me, it looks like one of those little soft squishy balls. And these capillaries are fenestrated, so they have lots of pores in them, and this allows for filtration. And remember, the fluid from filtration is called filtrate. And so what is filtrate? It is plasma-derived fluid. Plasma-derived fluid. Then the renal tubules are going to process this filtrate, and in the end we wind up with urine. So coming out of the glomerulus, we have filtrate. The second part is the capsule. I was taught Bowman's capsule, but now anatomy is changing everything from people's names to structures. And so it is like a balloon surrounding something on the inside. There are two layers, the parietal layer. Have you seen this before? Which is simple squamous epithelial, and the visceral layer. Remember, the visceral layer clings to the organ, and in this case, the visceral layer is clinging to the glomerular capillaries. And it has epithelial cells that are specialized called podocytes. Podo means foot, cyte means cell, so these are foot cells. And that's because these cells have extensions that cling to the basement membrane, and they're called foot processes. And in between the foot process are filtration slits that allow the filtrate to pass into the capillary space, also known as the capsular space. So let's look at a picture. So here we have the renal corpuscle, and you can see the glomerulus, the tuft of cells on the inside, and the capsule on the outside. And to the right, we have the different kinds of cells that we're going to find in each part of the nephron. So the glomerular capsule has a parietal layer, squamous epithelial cells. Then the capsule visceral layer is stuck to the capillaries proper in the glomerulus, and they share a fenestrated, that's where the podocytes are. This is a fenestrated epithelium. The next part of the nephron will be the renal tubule and the collecting duct. So the renal tubules are epithelial cells, but they're going to be different, and I'll point out differences to you. There are three major parts. There's the proximal convoluted tubule. It's called proximal because it's closest to the glomerulus. the nephron loop, which used to be called the loop of Henle, and then the distal convoluted tubule, which is furthest from the renal corpuscle and joins the collection duct, the collecting duct, not D-U-C-K, duct. So the PCT has cuboidal cells with microvilli, and these microvilli form a brush border. This increases surface area, and PCT is going to reabsorb and secrete. Reabsorb and secrete. And it is only going to be found in the cortex, and you can tell it apart from the distal because it is fuzzy on the inside. The nephron loop is a loop. It's a U-shaped loop. It has the descending loop that goes from the proximal convoluted tubule, makes a U-turn, comes back up, and joins with the distal convoluted tubule. So the proximal or descending loop, okay, simple squamous epithelium. The ascending loop is thick, and it usually has either cuboidal or columnar epithelial cells. The distal convoluted tubule has cuboidal cells, usually no microvilli. It functions to secrete more than reabsorb, so it does a lot more secreting. It is found only in the cortex, and when you look at a micrograph, you'll see that the proximal looks fuzzy in the center, but the distal is going to have a clear lumen, So here we are in the kidney tissue, and you see the big circular thing in the center is the renal corpuscle. Anytime you see this, you're going to think, renal corpuscle. So you should be able to define renal corpuscle and glomerulus. Renal corpuscle and glomerulus. When you look at the proximal convoluted tubule, this is the upper right corner, look down and you can see its center is fuzzy, and you can see a lot of fuzzy centers. And then just superior to that is the distal convoluted tubule, and look at the clear lumen. Look at the clear lumen. At the 11 o'clock position from the renal corpuscle, you can see another distal convoluted tubule. This one has cuboidal cells, and the center is clear. What about the collecting ducts? The collecting ducts collect. They have two types of cells, principal cells, and these cells are going to maintain water and sodium balance, and then the intercalated cells. Now remember when we talked about the heart muscle, we had intercalated discs, and these discs kept the heart muscle cells attached to each other, and they helped with producing the signal, keeping the signal going. In this case, the intercalated cells, A and B, isn't that nice that they named them A and B, are going to be connected to one another, and their job is to maintain the acid-base balance of the blood. And we'll talk about that when we talk about the physiology. Collecting ducts are going to receive the processed filtrate from a bunch of different nephrons. They're going to merge together and deliver the urine to the papilla and at the end of the renal pyramid into the minor calyces. So look at this picture again. Take time, find the renal cortex, find the renal medulla, find the renal corpuscle, find the proximal convoluted tubule. Then you see it runs into the descending loop. It makes a U-turn, comes back up to the ascending loop. Notice that the ascending loop is thick. The ascending loop merges into the distal convoluted tubule, and then that is going to connect to the collecting duct. And you see that the collecting duct has several different branches on it, and this is where distal convoluted tubules attach. Make sure that you study this and that you can identify each part. You can identify each part. What does convoluted mean? It twists all over. Twisty. So let's look at the two classes of nephrons. The cortical nephrons are mainly found in the cortex. Yay! The juxtamedullary nephrons have their nephron loops in the medulla. And this is important to produce concentrated urine. So when we look at this picture, you should be able to identify everything that's in dark print. Afferent arterioles, okay, efferent arterioles, vasa recta, peritubular capillaries. What is a cortical nephron and what is a juxtamedullary nephron? Juxta means next to. Next to. And then you have medullary in the medulla. So keep a look. Keep a look. What do you see here is that the afferent arteriole comes off of the cortical radiate artery. Cortical radiate artery. So now is the time to take a good break. Draw a line in your notes, stretch, come back in a couple of minutes, and let's finish this up. Here we go. There are two capillary beds, the glomerulus and the peritubular capillaries are going to be associated with the renal tubules. The renal tubules, glomerulus and peritubular capillaries. The juxtamedullary nephrons are associated with the vasa recta. You got to say that with an Italian accent. Vasa recta. Ooh, that makes me hungry for pizza. This is not good. Okay. The glomerulus, the capillaries, remember, filter the blood. So they're different from the other capillary beds because they're fed and drained by an arteriole. The afferent arteriole enters the glomerulus, and then the blood leaves the glomerulus by the efferent arteriole. The afferent arteriole arises from the cortical radiate artery. The efferent arteriole is either going to shoot its blood into the peritubular capillaries or the vasa recta. Okay, peritubular, these are going to be cortical nephrons, vasa recta, juxtamedullary. Got it? Okay. So blood pressure in the glomerulus is going to be high because the afferent arterioles are larger than the efferent, and so these arterioles are also high resistant vessels. Why do we want high blood pressure in these arterioles? Because we want everything we can to be pushed out of the blood into the filtrate. Remember, we're filtering. Same reason you use a vacuum when you filter in chem lab. You want to pull that fluid down through and filter it the best possible way you can. So you apply pressure. The peritubular capillaries, okay, are low pressure, remember. These arise from the efferent arteriole. They're going to be with the cortical nephrons, and then they empty directly into venules. The vasa recta is going to also arise from the efferent arterioles, but these are with the juxtamedullary nephrons, and they're very, very important in the function of forming concentrated urine. And this is because their nephron loop is very, very long compared to the cortical nephron. So here we have a nice 3D SEM showing the glomerulus, okay, the arteriole that supplies the glomerulus, and that's going to be what? The afferent, and then the efferent arteriole that drains the glomerulus. And you can see that the afferent is larger than the efferent. It's bigger around. So let's compare these two types of nephrons again. You have the cortical. Notice it has the short nephron loop, and it's mainly in the cortex. Then you have the juxtamedullary nephron, which has the very, very long nephron loop, and its loop is found in the medulla. Remember that the cortical have the peritubular capillaries, and the juxtamedullary have the vasa recta. The vasa recta. Whew. If you want, take a break. Draw a line. The juxtaglomerular complex, or the JGC. Each nephron has a juxtaglomerular complex. This is going to be modified portions of the distal ascending limb of the nephron loop, and usually the afferent, sometimes efferent, but mainly afferent arteriole. Juxta means side by side. So you've got an ascending limb of the nephron loop plus the afferent arteriole, and these are side by side close to the glomerulus. What's their job? They regulate the rate of filtration and blood pressure. There are three cells. You need to know these three cells. The macula densa, the macula densa, the JG cells. Your book calls them juxtaglomerular JG cells in one place, and then later refers to them as granular. All right? So these are smooth muscle cells of the arteriole. And so they're going to control how much blood flows through the afferent arteriole into the glomerulus, and they can sense blood pressure. They also contain granules for the enzyme renin. And remember, renin, angiotensin-renin system for controlling blood pressure. If you don't remember that, go back and review it. The third type of cell is called the extraglomerular mesangial cells. Extra, outside, glomerular, the glomerulus, mesangial cells. So these are in between the arteriole and the tubule cells. And we think that basically they pass messages back and forth between the macula densa and the JG cells. Note the term think. All right, so here you go. Here's the juxtaglomerular complex. You can see, all right, the afferent arteriole, the granular cells, or the JG cells, in the juxtaglomerular complex, and how it is side by side with the capsule, and then you can see the proximal limb. Okay, see the proximal tubule cells, and then the macula densa of the ascending limb of the nephron loop. Ascending means coming back up. Okay, so